So good afternoon, everyone. Welcome back to the CAFNR Research Council webinar series. This is the second and the last CRC webinar of the fall semester. And today we have a very special speaker, one of our colleagues, Dr. Pavel Somavat. And Dr. Jeb Vardhanabhuti, chair of the CAFNR Research Council, will make the formal introduction of our speaker. But before she does so, I would like to say a few words about the CAFNR Research Council, or CRC. We formalized CRC about 6 years ago as an advisory council that would provide advice to my office, that is, as you know, the CAFNR Office of Research. We do have faculty from every division represented on the council. We also have staff, grad students, and postdoc representation. This webinar is an example of some of the activities of the Council. In addition to providing advice to me and my office, they also organize, as we have noticed, the annual CAFNR Research Symposium in the fall, help make decisions regarding several research-related awards in CAFNR, and funding opportunities from my office, like the Joy of Discovery Seed Grant Program, the Matching Equipment Grant Program, and the Matching Assistantship Program. When they help make important decisions in the Office of Research, they are representing you. In other words, CRC plays an important role in shared governance in our college. And today, CRC is bringing another timely topic to you through the webinar. So we will have a Q&A session in the end, but you can post your questions in the Q&A box at any time. And Dr. Jeb will be moderating the Q&A session in the end. So without further ado, Jeb, the floor is yours.
Thank you, Dr. Jose. Good afternoon, everyone. Thank you for joining us. We are very pleased to have with us today Dr. Pavel Somavat, who will be presenting on leveraging the U.S. corn ecosystem for producing natural food colorants and value-added bioactive compounds. So before we begin, I would like to take a moment to introduce our speaker. Pavel Somavat serves as an assistant professor with a joint appointment in the Division of Food, Nutrition, and Exercise Sciences and the Department of Chemical and Biomedical Engineering here at Mizzou. He obtained a PhD from the Department of Agricultural and Biological Engineering, University of Illinois at Urbana-Champaign. His multidisciplinary research focuses on optimizing the extraction of anthocyanins and polyphenolic compounds from pigmented corn for innovative, value-added applications. This includes developing natural red colorants as sustainable alternatives to synthetic FD&C dyes and evaluating their thermal and storage stabilities. His group also advances sustainable soil processing and conducts techno-economic analysis of novel bioprocesses. His work has been supported by USDA NIFA, NCR SARE, the United Soybean Board, and the Good Food Institute. And now, please join me in welcoming Dr. Somavat. Dr. Somavat, the floor is yours.
Thank you very much, Professor Vardhanabhuti and Dr. Jose for giving me the opportunity. I also thank all the participants. It's later in the semester with so many things to take care. So with that being said, give me a moment. I share my slide. And so a quick question, can you see the presentation or not?
I think now we can see it, but it's not the full screen. It's not in the presentation mode.
Does it work now?
Yep, it works now. Perfect.
Okay. Thank you. Thank you so much. So again, as Professor Dr. Vartanabhuti mentioned, I'll be talking about our research work where we are utilizing colored corn varieties for natural food colorant extraction. So the agenda for the day will be, I'll be talking about synthetic and natural food colorants, why they are becoming a hot topic these days. Then I will introduce our work, which we are doing at University of Missouri, working to utilize the color corn varieties for the replacement of FD&C Red 40 and Red 3 dyes. I'll also talk about the Missouri Color Corn Breeding and Adaptation Program, which we are kind of running jointly with Dr. Sherry Flint Garcia from USDA ARS, with the aims of adapting colored corn varieties containing very high phytochemicals and anthocyanins and making them grow according to the Midwestern climatic conditions. In addition to the natural food colorant work, we are also utilizing the economically extracted phytochemicals for a number of other novel applications. Towards the end, I will open the session for questions and answers. So the role of colors in food. Now, the first impression a consumer gets of any food material is generated through visual inspection. And that's where color plays a very important indicator because it is a vital indicator of taste and flavor. To give you an example, if you see bananas on the right bottom, nobody will go for the darkened bananas, even though they might be more sweeter and good compared to a greenish banana. And that's the reason food industry typically goes for addition of or using vibrant colorants to make their products more attractive for the consumers. And the history of utilizing artificial food colorant actually goes back to the ancient Egyptians who used to color their candies. And we see evidence of Romans using iron nails to fortify red wines. So in order to make the food attractive, often appearance is given a way higher preference than the flavor of the food. Now, artificial food colorants are mostly used in the industry because of their lower cost. Now, I would stress that artificial or synthetic food colorants are synthesized from petroleum and coal tar, and they are preferred so far because of their lower cost, their higher stability, and we can make diverse and very vivid colors from petroleum or coal tar. The health concerns associated with synthetic food additives, which include colorants, actually are as old as their use in the food industry itself. And in 1975, Feingold actually reported that food additives, including food colorants and preservatives, may cause attention deficit hyperactivity disorder in children. Since the end of Second World War, the consumption of processed food has increased greatly. And with that increased consumption, actually, the consumption of synthetic food colorants in dye has increased fivefold to almost 67 milligrams of synthetic dyes per person per day. Now, in addition to the attention deficit hyperactivity disorder, various researchers have reported that synthetic food colorants may cause genotoxicity, neurotoxicity, and or carcinogenicity in humans. And more recently, FDA revoked the authorization of FD&C Red 3 dye to be used from food and cosmetics starting from 2027 because of the concerns associated with carcinogens. Now, this is not an isolated case. Actually, if you go back to the FDA website, since 1960s, dozens of food additives, including synthetic colorants, have been banned. So from time to time, FDA does steps in and bans. Well, at the same time, there is a movement at state levels in the US and where California is leading the charge. And last year, California actually banned the use of synthetic food colorants from their high school canteens. So there is definitely a push towards natural alternates of synthetic dyes. And I would also like to mention here that in 2010, European Union actually passed a legislation which forced the food industry to label the food items containing synthetic food dyes. And since then, the big food manufacturers, even though they may belong to the United States, they are producing food products in the European Union using natural colorants, but there has been no motivation for them to change to natural alternates here in the US. However, going ahead, we assume that things will not be the same because of the changing government priorities and increasing health concerns of the consumers. So with that being said, actually, there are colors in nature which we humans have been consuming. So if you think of dark colored fruits and vegetables containing carotenoids, anthocyanins, betalains, curcumin, chlorophyll. So the gist is we humans have been consuming natural food colorants, but the main problem is they are expensive at the front end. Sometimes they don't produce as vivid color as the synthetic ones, but there is definitely a possibility. Now, anthocyanins are the natural pigments which are responsible for the red, blue, black, and purple colors of the nature, and they may replace FD&C Red 40 and Red 3 dyes. It is worth mentioning here that Red 40 is actually 40% of all the synthetic food dyes in the US. So it's a very big or considerable market. Now, on their basic level, anthocyanins consist of anthocyanidins, which contain two aromatic rings bonded to a heterocyclic ring containing oxygen, and their glycosidic forms are known as anthocyanins. Depending on the sugar moiety, anthocyanins may have different chemical configurations, and the resonating structure of flavulinium cation exhibits different colors of anthocyanins. So if you look at the bottom, even the fall colors are actually caused by anthocyanins. So in addition to their use as natural food colorants, anthocyanins are very powerful antioxidants and anti-carcinogens. So if you think of health promoting benefits of red wine, actually many of them come due to the presence of anthocyanins. Blueberries, on the other hand, we eat blueberries because of their high anthocyanin and polyphenol content. So the gist is anthocyanins can be used as a replacement of FD&C Red 14, Red 3 dyes, and at the same time, you can actually supplement the product due to the health promoting benefits of the anthocyanins. Now, when it comes to the European Union, the current approach they have taken to get natural red food colorants is they're using dark grapes, dark carrots, or red cabbages to actually extract the colorants. Now, these approaches use solvent-based extraction methods, or sometimes they need to do juice concentration. However, if you think in terms of the raw material used for anthocyanin extraction, they are expensive on the one end. And then if you think of blueberries as a source of anthocyanins, they typically contain 1 to 2 grams of anthocyanins per kilogram of blueberries. And if you think of extracting anthocyanins as a colorant, the whole material needs to be macerated, and then you need to come up with proper solvent system to extract those colorants. What it means is the processing residue in after post-extraction doesn't have any economic value. On the other hand, if you think of dark carrots and red cabbage as a source of anthocyanin, their juices are concentrated, but down the line, they require further purification to take off the volatile aroma and taste factors which may influence the final products. So all these problems actually make the use of anthocyanin expensive, and that's where some of our research with color corn varieties kind of serves to answer those questions. So the hypothesis is we have, I mean, last 30 years, researchers have identified that colored corn varieties, which are native to the highlands of South America, contain very high amount of anthocyanins and phytochemicals. So our hypothesis is, since here in the US, we are so good at growing corn and processing corn, so why not we explore the possibility of utilizing colored corn varieties to extract natural food colorants? So in that respect, I'll briefly talk about the U.S. corn industry. Although this figure is a bit old, but the numbers remain the same. So I went with it. So when it comes to the corn usage, typically 15 billion bushels of corn is harvested in the US every year. And a lot of it is utilized, and in fact, almost 40% of it is utilized by the what we call corn dry grind industry, which converts corn into ethanol, and then it ends into our gasoline tanks. So almost 40% of corn we harvest every year ends up as a low value fuel additive, and there is a very minimal proportion of value addition. However, because of government subsidies, we have been able to sustain that thing. In addition to that, I will briefly talk about the corn milling industry. So dry milling industry is the smallest in terms of volume process. It processes about only 1.5% of the total corn harvested every year. And this industry mainly utilizes corn for producing food products such as breakfast cereals, grits, which are used for making Cheetos and other products, corn flour and corn oil. Now, the second biggest industry is corn wet milling, and it typically processes 1.7 billion bushels per year. So wet milling involves a chemical refining of corn to extract the chemical constituent, and prime products are starch, which is then converted into high fructose corn syrup, flavors, enzymes. And actually, wet milling industry contributes to almost 1,000 different industrial products, which are used in U.S. industry. Now, the contribution of U.S. wet milling per year to the economy is almost 71 billion US dollar. Dry grind industry, again, is the minimal in terms of value addition, but it is by far the bigger consumer of the corn. And this process is oriented towards biofuel production. Now, even though dry grind industry processes more than 3 times the corn processed by the wet milling industry, the contribution or the financial contribution is just 46 billion, which is 65% of wet milling industry's contribution. So the gist is there is not enough value addition when it comes to corn utilization. And going ahead, there will be challenges because right now, even though corn farmers and processors have ethanol industry kind of serving the gasoline demand of the US, but if we think of going ahead in future with more and more adoption of electric vehicles, it will become a problem. So it is high time for corn farmers as well as processing industry to look for value added proportion when it comes to corn farming and utilization. And that's where some of our work comes into play. So as I mentioned, the color corn varieties native to the Southern America, they actually contain a lot of anthocyanins. So blueberries and dark grapes typically contain anywhere from 1 to 3 grams of anthocyanins per kilogram of blueberries or dark grapes. But in some of the color corn varieties, actually we have quantified as high as 5 grams of anthocyanins per kilogram of corn. So it's almost 4 to 6 times as high as compared to some of the high health promoting fruits and vegetables. Now, that's only one part of the story. During my doctoral research at University of Illinois, we actually worked with color corn varieties, and we figured out that most of these polyphenolic compounds in color corn varieties are actually concentrated in pericarp or bran, which is the outermost layer of corn kernel. So if you see at the pictures at the top, these are microstructure images of the corn kernel being dissected. So the color you see at the top is the pericarp layer, which is the typical layer which gets stuck in your teeth when you eat sweet corn. So for corn industry, actually, this is a waste material because we humans cannot digest fiber. And in case of purple corn, all the phytochemicals and anthocyanins are concentrated in what is a waste for corn processing industry. The corn processing industry is mainly interested in endosperm, which contains most of starch and protein. So actually, during my doctoral research work, we were able to separate these phytochemical rich pericarp at the front end of the corn processing and then selectively utilize this for polyphenol extraction. And actually, in our studies, we quantify almost 35 grams of anthocyanins per kilogram of separated pericarp. So herein comes the value-added proposition. If you think of blueberries and dark grapes, not only they contain a relatively lesser amount of anthocyanins and phytochemicals, in order to extract, oftentimes you have to deal with the whole material, which is expensive, and the processing residue doesn't have any value. However, in case of colored corn varieties, we are able to recover the phytochemical rich pericarp at the front end of the process, which means the corn endosperm containing all the starch and protein can be utilized by either dry grind or dry milling industry. Whereas on account of its very high anthocyanin and polyphenol concentration, pericarp fiber can be utilized as natural food colorant, health supplement, or pest deterrent. So just to show the value. Now, when I joined University of Missouri, I was basically building on our preliminary research, which we did at University of Illinois. So one of the questions was, even though we were able to quantify almost 35 grams of anthocyanins per kilogram of pericarp, there was still open questions. Can we further optimize the extraction? So initial part of our research, or one part of our research, focused on polyphenol optimization, extraction optimization from pericarp using advanced assisted techniques and solvent systems with an aim of reducing the overall time and improving the extraction efficiency. So we use microwaves, we use ultrasounds, we played with different solvent systems. Since we had food grade applications in mind, we were always using environmental friendly solvents. And actually, in some of our research using ultrasonic extraction and microwave extraction, we actually improved this extraction to almost 39 grams of anthocyanins, 138 grams of polyphenols, 76 grams of flavonoids, and almost 280 grams of condensed tannins per kilogram of separated pericarp. Now, if you think on a weight by weight basis, there is nothing in this world so rich in terms of anthocyanins and other polyphenolic compounds. And the advantage that comes with corn, if you think of blueberries and growing other high value product, so we see definitely a great opportunity. The next question we ask ourselves was, okay, we can use these colored corn varieties to economically extract natural red colorants as well as other health promoting phytochemicals. But can we grow these varieties here in the US? That's where Dr. Sherry Flint Garcia, USDA ARS geneticist and a corn breeder, we were able to kind of connect with her. She was already interested in growing some of these, what we call as heirloom varieties. So we started in 2021. We requested her, and actually she shared 14 colored corn varieties, which she grew in 2020 here at University of Missouri Farms. So we processed those varieties and we compared their polyphenolic contents with purple corn or maize morado, which is a native of South America. So the results from our first evaluation we published in Industrial Crops and Products. So what you see here is in the graph on the top left is the total anthocyanin content of maize morado or purple corn, which is the control or the South American variety. And actually, when Dr. Sherry used maize morado as a parent to adapt some of the existing color corn varieties here in the US, even in the first and second iteration, we saw that there are varieties which contain very high amounts of anthocyanins, phenolic acids, as well as flavonoids, and we can actually grow them here in the Midwest. So we went back to Dr. Sherry, we shared our analysis, and she used that information for the next breeding cycle. That was in 2022. Just earlier this year, we published this work. So she shared another 15 varieties which she harvested in 2022. And again, we kind of quantified, as you can see from the pigmentation of the kernels, we demonstrated that very high polyphenol containing varieties can actually be grown in Missouri. Now, in 2023, we again got varieties from the third iteration. This time, if you see the kernels, they were not very dark, but in fact, they were very colorful. So as engineers and processors, we were disappointed. I went back to Dr. Sherry and said, Dr. Sherry, I was expecting more pigmentation, but this time we don't see much darkness. She laughed and she said, the problems of a breeder is different. We go for diversity. So what she did in third iteration was she played with different native cultivars. And actually, when we analyzed these varieties, we saw interesting differences. So if you see the selected anthocyanin concentration of 5 best varieties out of what we got from 2023, the anthocyanin concentrations are not as high as what we saw compared to the earlier years. But when we did HPLC analysis, the differences became clear. Actually, in terms of diversity of anthocyanins, flavonoids, and phenolic acids, these varieties actually contain very vast diversity of these compounds. And actually, that is not a bad thing, because if you are going with different hues of red color, probably you are not just wanting one kind of corn. So even though at the beginning we were disappointed, but down the line we see very interesting opportunities associated with this breeding. And then again, here is the metabolomic analysis of one of the varieties. And actually, we quantified 74 bioactive compounds, including 30 flavonoids, 11 different types of anthocyanins, and 19 phenolics. So the gist is we can produce very high yielding varieties here in the Midwest. Now, another question we asked was, okay, we have the corn varieties. Can we prepare shelf-stable food colorants for them, from them? So we actually use color corn varieties. We use spray drying to make shelf-stable powders. We did physical characterization, and at the end, we actually demonstrated that you can use these color corn varieties to make free-flowing shell-stable colorant powders, which retain very high amounts of total anthocyanins, condensed tannins, flavonoids, and phenolics. Going ahead, the next question we asked ourselves was, will these colorants prepared from color corn varieties be stable in finished food products? Because that's the next question industry might ask us. So we further built upon our research, and we, based on the previous powder formulation experiments, we actually used pericarp extract. We formulated spray-dried and freeze-dried powders, colorant powders, and then we introduced those colorant powders into model beverage system at 3 different pHs, 3 different ascorbic acid or vitamin C concentrations, and we exposed them to 3 different temperatures over a period of 7 weeks. In addition to that, we also exposed our model beverage system to 4 temperatures, 50 degrees Celsius, 70 degrees Celsius, 90, and 121, to study the effect of thermal processing. In case we introduce these colorants to, let us say, a finished product, will the anthocyanins and the colorant last or lost the heat treatment. So the results were very interesting indeed. So on the left, you see the uncoated extract where we just use the liquid anthocyanin or colorant extract and spray dried it. Then we have spray dried extract with 1% maltodextrin, and third was a freeze dried extract with 1% maltodextrin. So we studied the anthocyanin degradation kinetics, and what is important is I'll bring your attention to the half-life at temperatures of 121 degrees Celsius. So even when the model beverage system containing anthocyanins were exposed to temperatures as high as 121 degrees Celsius, it was almost 2.5 hours it took for half of the anthocyanins to degrade. So actually, these color corn anthocyanins are very highly stable. So this research work clearly showed that not only these colorants can tolerate the thermal treatment, in fact, they are very, very stable. So in another experiment, we actually purchased FDNC Red 40 dye, and we used our 3 treatments and exposed these systems to 121 degrees Celsius over 1 hour. And we took images at every 10-minute interval. So you can see synthetic red dye after 1 hour exposure to 121 degrees Celsius, you don't see much change in color. And actually with all 3 samples, uncoated, spray dried, and freeze dried, our colorants were equally stable. In addition to that, in the model beverage system, when we stored our beverage system at 4 degrees Celsius, the half-life of anthocyanins were 18 weeks. If as same 40 degrees Celsius, it's a very high temperature. Still, they had a half-life of almost 2.5 weeks. Different pH. So the gist is we show the color corn varieties can be used to make very highly stable red food colorants. Now, during this breeding program, we actually saw some brown varieties, and at the same time, the caramel food colorants, which are made from burning sugar, are being implicated to have 4-methyl imidazole, which is a potential carcinogen. So actually, we also see an opportunity of utilizing some of these brown color varieties to make caramel food colorant. So that's also one thing we are looking into. And in addition to that, so we are actually utilized. So if you think of growing color corn varieties here in the Midwest, maybe a finite amount of harvest would solve the problem of natural red food colorants. Then the question farmers and industry may ask us is, okay, we may go with color corn varieties, but what could be some of the other applications? And that's where things get very interesting. Since we can economically extract these polyphenolic compounds from color corn varieties relatively inexpensively, they can be actually used for many different applications. And one of the applications we have been working with is, since secondary plant metabolites are synthesized by plants due to a reason, and plant defense is one of the things. So we actually used very highly concentrated phytochemical powders, and they demonstrated antimicrobial effect against 2 gram-positive and 2 gram-negative strain of bacteria. So we actually brought in these highly concentrated phytochemical extracts and made protein-based biodegradable films. We incorporated these antimicrobial effects, and now we see their application as edible antimicrobial and biodegradable food packaging, which can extend the shelf life of packaged food products. And you can even eat those products. So sausage packaging could be some of the applications. Another application we are utilizing these phytochemical extracts is we are using edible coating and kind of fortifying those edible coatings. And we already have completed a study where we were able to extend the a shelf life of strawberries to almost 14 days using edible food coatings fortified with phytochemical powders. On the other side, one of the maximum avenues for value addition with corn is obviously bourbon making. Now, these color corn varieties contain phytochemical compounds, so there is definitely an opportunity where we can manufacture specialty alcohols with color corn varieties where due to the presence of polyphenolic compounds, we may have unique color and aroma profiles of those alcohols. Lastly, separated pericarp is a mix of dietary fiber and polyphenolic compounds. Now, the Americans on an average only eat 60% of recommended daily intake of fiber. So one avenue we are working on is to make separated pericarp powders and then incorporate those powders in different food formulations with an aim of kind of not only increasing the fiber content of the food product, but at the same time, we bring in anthocyanins and other polyphenolic compounds. So many researchers have already shown that the intake of anthocyanins kind of reduces the all-cause mortality. Now, the average per person intake of anthocyanins in the US is 10 milligram per person per day. And if you think of 1 gram of pericarp, it's almost 3.5 times greater than that. So if you are fortifying, let us say, a pancake or a muffin with 5 grams of pericarp, now you are bringing in anthocyanins, which are twice greater than a glass of red wine. So again, there is an excellent opportunity to fortify different food products with fiber coming from this pericarp. Again, I hope I'm able to give you an idea what could be some of the excellent opportunities associated with these colorants. So lastly, I'm sorry I may have run over time, but I would like to thank my grad students who do all the hard work, as well as our team. So Dr. Sherry Flint Garcia, she does all the hard work on the field and gets us these colored corn varieties. And then again, the students. I also thank Dr. Ellen Vaughn from Bioengineering. We have collaborated a lot. And lastly, I would like to thank USDA NIFA and NIFA NCR Sun grants, which we have been able to utilize for some of the research work. With that being said, I thank everyone for your kind attention and will be happy to answer any questions.
Hey. Well, thank you very much, Dr. Somavat. So we'll now move into our Q&A session. So again, if you have not already, please submit your questions using the Q&A feature, and we'll get through that as many as we can. I actually have a few questions myself. So do you think well, this related to the work, Dr. Sherry, do you think there would be more selective breeding project that enhance corn anthocyanin?
Yeah, that's a very good question. Actually, so far, what we have been doing is to identify the best varieties, and then she's using that variety as a parent. So she's not using any genetic modification. So it takes time, and every year we are improving. But we do see diverse varieties. We do see some varieties which contain only a limited amount of anthocyanins, but they are darker. But we also see diversity in terms of colors, as I showed in the third iteration. So there, we foresee interesting opportunities. But then again, the question you may ask is, what about the yield? Unfortunately, we are not there at that point. We hope with the changes taking place in the government, we hope that we get further funding support to be able to kind of now start focusing on some of the selective varieties and do yield related research. So I hope I answer your question. We are making a steady progress, but we need a bit more time because it's not genetic modification. It's using traditional techniques, and the iterations need to go up. And
So are there any other groups that working on purple corn?
no,
United
I
States?
mean, we have the advantage of being the only group here in the US which is actively engaged not only in processing as well as in adapting the color corn varieties. So that's the advantage we have at University of Missouri.
Okay, but Dr. Sherry is not interested in genetic engineering, right?
No, because
Okay.
then it defeats the purpose of getting value-added products. And
Okay.
her interest, actually, before I came here, she is a very dedicated corn breeder. So she thinks about the populations, and
Right.
she's not into genetic modification.
I see Dr. Joe's. That's a question.
Yeah. So let me ask you a quick question, Pavel. Excellent job. So I know we have talked about this before, but how do you work with industry or convince industry to adopt, your purple corn colorant and some of the food packaging and other ideas that you discussed here? How do you get it into the hands of industry? So this is going to be in the marketplace.
Yeah, so far I must confess that that has been my Achilles heel. I'm good at doing work in my lab, not that good going out, and I must confess. But I assure you, I'm trying my best. Uh, uh, 2 weeks ago, actually, I had an interaction with the scientists from PepsiCo who are interested in some of what we are doing. But going ahead, I do see with the funding becoming a problem. I will be forced to step out of my comfort zone, and I'm trying that, I assure you. And I'll welcome any help if you can render me, because then again, you are more connected. So I'll appreciate any help from outside as well.
Okay. All right. Thank you.
Have you patented this, right? Or not yet?
Actually, the original process of separating the pericarp, that was my doctoral work at UIUC. That constituted a novelty in a way that we were able to do that. But most of the other work is more application in nature, and that's where, even if you think of investing in patents, at times you might come across problems where forcing your patent might be an issue. So I think it's more or less applied work. But if we can, Professor Jose is very correct in terms that if we can bring in industry, we can, with the copyright and those kind of things, kind of licensing agreements, that will be a more rational approach of using some of the applications we are developing.
Okay. I actually have another question. Maybe perhaps one last question here.
Sure.
In terms of like testing, any other testing to get the approval from the FDA?
So that's a good thing with this corn, where we are extracting the colorant is an edible part of the kernel. So it is GRAS. That's the problem. It could be a problem for many other things, but we are extracting something from the edible part of a product. So that may not face, because we do eat corn kernels. So if I'm just recovering pericarp, separating it constitutes GRAS. So I don't foresee a lot of problems. It's a very good question, but yeah, we have the advantage.
Okay. Well, thank you very much, Dr. Somavat, for this colorful and engaging presentation.
No, thank you very much for giving me the opportunity, and I greatly appreciate this.
And Dr. Jose?
All right. Well, let me also add my thanks to Dr. Somavat for an informative talk and for responding to our questions. And thanks to all of you for joining today's webinar. And as I said earlier, this is our last webinar for the semester. So we will see you back here at the webinar in the spring. Thanks, everyone.